next speaker, which is paper 01C, spectroscopy of lithium atoms using an optical frequency comb. This represents research performed at Oberlin College, and our speaker will be Donal Sheets.
Hello, I'm Donal from Oberlin, and I'm here to talk to you about a project I've been working on for a while with my advisor, Jason Spong. This is precision spectroscopy of atomic lithium. So the first question is, why is lithium interesting? Lithium is a fairly simple atom, being a 3-electron atom. You can see it's the third atom in the periodic table. And so this means that it is possible to solve the energy equation using an extension off the way you solve hydrogen, which is the Schrodinger equation. So we take the Schrodinger equation, we add in relativistic corrections and quantum electrodynamics, and we have our prediction for the energy levels in the state. So this is important as we are testing how these corrections are added. We're not testing QED, we're testing how they're added into these calculations. Lithium also is a window into nuclear theory, as it has two stable isotopes, lithium-6 and lithium-7. This means that mass and volume changes allow us to look at how those affect the energy levels and how they shift. So what we're looking at is two transitions in lithium, the so-called D1 and D2 transitions, which are the first excited state. These are shifted by 10 gigahertz, which we call fine structure splitting. We're also looking at the isotope shift between lithium-7 and lithium-6. This is also about 10 gigahertz. Notice this is about the same as the fine structure splitting. So the current state of this measurement is that there's a wide discrepancy throughout the experimental values. Each point here represents a different group's published value of the measurement. This is two examples of the transitions we are measuring. It is not. So you can see that the red points are all experiment, and that one purple point is the theory. Note that most of the points do not agree with the theory, and they obviously don't agree with each other. This is caused by difficulty in evaluating systematics, as well as other issues incorporating various effects. So how do we make these measurements? We use fluorescence spectroscopy, which uses a laser to probe our atoms. So the laser light comes in, and if the laser is on resonance with the transition, the atom absorbs energy from the laser and goes up in energy with the state, and then it decays and emits light, which we can detect. Note that the transition is frequency dependent, and so we are scanning the laser over the transition and hope to see these peaks of fluorescence dependent on our frequency of our laser. So now that we know where our transition is, the important part is figuring out what's the frequency of the laser, and that's actually the hard part. So how do we do this? We use a femtosecond frequency cone, which is what could be called a white light laser, meaning it has over 100,000 different frequencies spanning the visible spectrum. So this means that the entire spectrum is contained in one laser. This is important as it allows us to measure the frequency of our single mode laser by referencing it to this cone. The cone, each tooth of the cone is spaced by a given microwave frequency called the rep rate. This is a spacing between each tooth of the cone and is constant throughout the cone. Combined with an offset frequency and a mode number, this allows us to relate any optical frequency to a pair of microwave frequencies, which are easy to count. Optical frequencies very difficult to measure. So this effectively allows us to synthesize optical frequencies to microwave frequencies. So this is our frequency cone. It is a high sapphire laser, meaning it is a sapphire crystal doped with titanium. It is stabilized to a GPS-guided rubidium atomic clock. This allows us to relate our optical frequencies to the SI second standard. This gives us very high frequency accuracy and will allow us to get a very precise measurement. So here you see in the top, this is our laser cavity. These are some of the optics used to control it. And then this is the output of the cone off the diffraction grating, so you can see the full spectrum of light there. So our experimental setup, we use an atomic beam source from a lithium oven, which we then excite with a diode laser. This diode laser is then referenced to our frequency cone, which we use to measure the frequency of the laser. Each microwave frequency of the cone is referenced to the clock, and this allows us to stabilize the cone to the clock and then the laser to the cone, giving us our stabilization. So a little more on our experimental setup. This is the atomic beam out of the oven here. This is our interaction zone. There's the PMT is up here, which we use to measure our fluorescence of our atoms. And then this is our homemade diode laser. So the current status of this experiment is that we've successfully constructed all the apparatus. It all works. We're working on collecting data. This is a preliminary plot of a couple transitions, and we just need to work on evaluating systematics to really figure out what shifts are present and what we need to improve. So future plans, once we get these transitions done, we want to look at some higher lying states. You can see here these are the two transitions we've been looking at, D1 and D2. And we can access some of the higher states using a combination of cone light and diodes. We also hope to cool and trap the atoms, which will allow us to get to states which we can't currently. Some acknowledgements. I'd like to thank my advisor, Jason Stoneker, as well as some of the current and past students in the lab. A little plug, Peter is giving a poster in the session this afternoon. If you want to hear more, you should go talk to him. This work is supported by the NIST Precision Measurements Grant as well as the MSF.
Questions for Donal?
So you showed that very nice preliminary line profile, and I just wondered, could you flip back to that? Just wondered, what kind of next analysis would you be doing on that? Would you be, yeah.
So currently this data was taken without the diode locked to the cone. So that's why if you notice this is relative frequency, not absolute frequency. So there's other ways of calibrating it which don't give as high precision as locking it to the cone, but are easier to do in the lab. So when you're evaluating some other systematics, it's easier to do it just using that. So we have some data that's locked to the cone. We're also trying to limit background noise and limit things such as we need to evaluate the magnetic field in the interaction region. We also need to evaluate Doppler shifts, so we're working through that and sort of nailing down the systematics.
So on that note, with the systematics, is that the goal for laser cooling and trapping then? Would be to try to eliminate some of the systematics that go into the measurements?
Yes and no. It allows us Doppler-free measurements, so that's a big plus. At the moment, we have a line width from our atomic beam of about 25 megahertz, and the transition line width is like 6 megahertz. So we can make a measurement to the accuracy. We hope to make a measurement to the accuracy we want with the atomic beam, but it would be nice to not have to deal with that.
Is that just limited by the expansion of the atomic beam?
Yes.
Okay, we have
time for one more short question, if someone would like. Okay, not, it's time for a speaker again.